Hello, my name is Víctor Díaz. I am a student of PhD in biochemistry in the University of Chile, and it is a pleasure to speak you about our investigation called Evaluation of a Novel Tool for the Study of Early Metastasis, based in B16F10 murine melanoma cells labeled with cadmium tellurium quantum dots. Nowadays, cancer is one of the main motives of death across the world. Tumor cells dissemination to distant organs, known as metastasis, is the major cause of death related to cancer. However, metastasis is a process which evolution in vivo is hard to follow, mainly by the low number of cells present in bloodstream that reach and colonize distant organs in a clinical detectable way. Traditional study of metastasis involved the administration of metastatic cells into the bloodstream of the animal model. After a couple of weeks post-administration, necropsy is performed post-euthanasia for the evaluation of metastasis in organs. As an example, in our laboratory has been using the Syngeneic model of melanoma cells, B16F10 cells, and C57BL6 mice for in vivo metastasis study. With this model, we can measure the metastatic capacity of B16F10 cells after 40 days post-injection. But what happened between these 40 days is still a mystery. 2 years ago was reported that the accumulation of melanoma cells in lungs of C57BL6 mice is a process that starts at 5 minutes post-cell injection, making clear the need of tool of an imaging technique that permit allow the process of migration and invasion in vivo. For this reason, the in vivo metastasis study can be described as a black box study, since it's not possible to observe metastatic cells movement or the accumulation of these cells in different organs of animals. That's why in vivo cell tracking is urgently required to diminish the metastasis assays time and the need of development the methodology to obtain labeled cells that can be used in metastasis assays. Several studies propose the use of quantum dots as a method for labeling cells and tumors. Quantum dots are fluorescent semiconductor nanocrystals, and these nanoparticles are formed by a central core consisting of atoms of different elements like cadmium, lead, tellurium, etc. This quantum dot can emit fluorescence at different wavelengths depending on its size and composition. Compared with organic fluorophores, quantum dots possess improved properties, high stability, high brightness, high fluorescent lifetime, and the possibility of bioconjugation of this quantum dot with different molecules, and the use of these nanoparticles for multispectral analysis. Quantum dots have been used in vivo imaging assays, but the use of these nanoparticles has been limited by their toxicity in biological systems associated to rust generation and heavy metal release. As a solution of this problem, our research group previously reported a new quantum dot synthesis method called biomimetic. This method is easy, fast, economic, and eco-friendly. Biomimetic quantum dots were used for cancer cells labeling, and it was observed that the internalization of this quantum dot does not affect viability or morphology of cancer cells MKN45, but still is unknown the possible effect of this quantum dot's internalization of the metastasis capacity of cancer cells. Based on this antecedent is that we hypothesize that B16 stem cells labeled with biomimetic quantum dots serve to monitoring early stage of metastasis or metastatic cell accumulation in organ of C57BL6 mice. To prove this hypothesis, we propose the use of B16 stem cells labeled with quantum dots, which will then be used to characterize the effect of the internalization of these quantum dots in its viability, proliferation, migration, and invasion capacity, along with modulate the possible effect of this quantum dot internalization by treatment with N-acetylcysteine, to finally determine the method that allows us to obtain B16 stem cells that can be used in vivo or ex vivo metastasis assays. The quantum dots used were synthesized using the biomimetic method, and then their size, charge, spectroscopic characteristics, and cadmium and tellurium amount present in quantum dots were characterized. Then, these quantum dots are used to labeling B16 stem cells following protocols described by Pérez et al., plus NAC treatment to modulate the effect of the internalization of quantum dots on the B16 stem cells metastatic characteristics. Subsequently, the viability, the internalization of quantum dots, and those level of labeled cells in presence or absence of NAC were determined. Then, these cells labeled in the presence of NAC were used in invasion, proliferation, and migration in vitro assays in order to determine the method to obtain B16 stem cells labeled with quantum dots, which eventually can be used in assay for metastasis in vivo or early metastasis in vivo cells. First of all, we perform the characterization of the quantum dots. The data show that the synthesized quantum dots have approximately a size of 5 nanometers and a negative charge of approximately 30.6 millivolts. These quantum dots emitting a red fluorescent with a peak of 519 nanometers and are composed by approximately 29% of cadmium and approximately 20% of tellurium. To evaluate the incorporation of this quantum dot into B16 stem cells, these cells were treated with different concentration of quantum dots. Data show that the treatment with 50, 100, and 200 micrograms per ml of red quantum dots and lipofectamine do not affect the viability of B16 stem cells. The biggest male fluorescent intensity, or MFI, it was obtained with red quantum dot treatment at 100 micrograms per ml of quantum dots. In this picture, we can see B16 stem cells treated with live-ran cell tracker, which ran viable cells at green, and cells treated with live-ran cell tracker and quantum dots. This data shows that the treatment used allows the viable B16 stem cells obtain cells labeled with red quantum dot inside. It's well known that the presence of quantum dots produces an increase of ROS levels in different kinds of cells. A graph showed that the internalization of quantum dots increased the ROS level. This could reproduce change and characteristics of these cells. By this reason, the cells were treated with NAC. NAC decreased ROS level into the cell. Interestingly, NAC treatment significantly increased the percentage of labeled cells almost to 100%, and the MFI related to the amount of quantum dots inside cells. This result suggests that the NAC treatment decreases ROS levels and increases internalization of red quantum dots in B16F10 cells, and the treatment with 4 millimolar of NAC increased the percentage of viable labeled cells. On the other hand, treatment with 10 millimolar of NAC increased the percentage of viable labeled cells and the MFI, and this really is related to an increase of the amount of quantum dots inside the cells. To compare the effect that produced the different treatment performed in metastatic capacity of B16F10 cells, quantum dot labeled cells in presence of 4 millimolar of NAC and 10 millimolar of NAC were used to perform migration and invasion in vitro assays. The figure shows in red circles the migrated cells and in white arrows the fluorescent related with quantum dots. This figure shows that the B16F10 cells, labeled cells, are able to migrate. When migration was quantified, we found that the internalization of quantum dots affects the migration. Treatment with 10 millimolar of NAC allowed to obtain B16F10 cells labeled with quantum dots, and these cells can migrate in a manner similar to the control. This data suggests that the NAC treatment allows modulate migration in quantum dots labeled cells mediated by ROS, and propose a protocol allow for cells labeled with quantum dots that migrate similar to the control. Later was performed an in vitro invasion assays using B16F10 cells labeled with red quantum dot in presence of 4 and 10 millimolar of NAC. The figure showed that the B16F10 cell labeled are able to invade. Surprisingly, when invasion was quantified, we found that the internalization of quantum dots significantly decreased the invasion of these cells, and this decrease was related with the treatment with NAC. This data suggests that the internalization of red quantum dots decreased invasion of B16F10 cells. Based on these results, it was decided to use quantum dots labeled cells in the presence of 10 millimolar of NAC for the next analysis. And now, these labeled cells was called as labeled cells. To determine the labeling persistence into B16F10 cells for posterior application in vivo metastasis assays, labeled cells were cultured during 5 days after its labeling, and the total source numbers, cell viability, viable labeled cell percentage, and mean fluorescent intensity were determined. The result shows that the labeled cell's viability was not significantly impaired during 5 days post-labeling, but a significant reduction of labeling was observed from the 4th days, and a significant reduction of MFI was observed from the 2nd day. Interestingly, labelled cells appear to be incapable to proliferate, since no increase in the total number of cells was observed during the 5 days of the assays. This data suggests that the red quantum dots internalization does not affect the viability of cells even after 5 days post-internalization, and the red label decreases from the second day, and finally the internalization of these red nanoparticles inhibit the proliferation of B16F10 cells. In summary, the treatment with this quantum dot in the presence of 10 mmol of NAC favored in the internalization of quantum dots into B16F10 cells. It's possible to obtain labeled cells with the same capacity to migrate that unlabeled cells, and keep these labeled for at least 2 days. Labeled cells can invade in a smaller amount compared to the control cells and are unable to proliferate. For this reason, we believe it is possible to use these metastatic cells for follow their arrival in different organs of C57BL6 mice. Later, an in vivo characterization and tracking of the metastatic ability of B16F10 cells labeled with quantum dots was performed. In this study, it was used a Syngeneic metastasis model to evaluate the metastatic capacity of B16F10 labeled cells. These cells was injected into immunocompetent C57BL6 mice by tail vein. 21 days post-injection, mice survival and tumor mass in lung were determined. Data show that the B16F10 labeled cells administration does not alter mice survival. After 21 days, mice necropsy showed that approximately 92% of the B16F10 labeled cells treated mice did not exhibit metastasis. This result suggests that the internalization of quantum dots in these metastatic cells inhibits its metastatic ability. To assess the potential of this method as early metastasis assays, labeled cells were injected into C57BL6 mice tail vein for their ulterior in vivo fluorescent tracking. In figure A showed that it's possible to detect in vitro the fluorescent emission at 600 nanometers of labeled cells when excited at 400 nanometers. Later, after necropsy, organs were collected into fecal solution for organ decoloration and preservation. Figure B shows image obtained by in vivo imaging system of C57BL6 mice lungs obtained after 6 hours inoculation of labeled cells. A distinguished signal associated to quantum dots labeled cells was obtained after organ fixation. Lung histological slides from mice inoculated with labeled cells showed fluorescent emission concordant with red quantum dot signal. A result that was not observed in the control. This result suggests that it could be detected fluorescence associated to red quantum dots by imaging tools, and this fluorescence can be related to the presence of B16F10 cells labeled in the lung. Finally, to tracking the early cell accumulation process, an accumulation kinetic was performed in C57BL6 mice organs. For this purpose, after the administration of labeled cells into mice vein tail, the following organs were analyzed: lung, the metastatic cell's target, heart, kidney, and liver. Mice necropsy and organ harvesting was performed after 5 minutes, 20 minutes, 2 hours, 6 hours, and 24 hours post-injection. The fluorescence quantification observed by ex vivo imagenology of the organ showed that the greater difference was observed in lung harvested after 6 hours post-labeled cell injection. Figure A showed a significant fluorescence increase after 5 minutes post-injection, which kept growing until its maximum at 6 hours post-injection. At 5 minutes, it also observed an increase in heart, kidney, and liver fluorescence. Nevertheless, afterward, kidney and liver fluorescence intensity diminished to values similar to control organs. It's possible to observe that the heart fluorescence which increased, but to a lesser extent than lung. This data showed a specific positive lung tropism and that it's possible to detect fluorescence associated to quantum dots labeled cells by imaging tools in different organs of C57BL6 mice. These results suggest that it's possible to detect the fluorescence associated to B16F10 cells labeled by imaging tools in different organs of C57BL6 mice, and as expected, the large increase of fluorescence associated to B16F10 cells occurred in the lung of C57BL6 mice. This intensity increased over the time with the peak of 6 hours post-injection. As a conclusion, in this study it is described for the first time the effect that quantum dots incorporation into B16F10 cells exert, and a novel methodology that allowed to modulation of quantum dot internalization effects. The quantum dot internalization affects metastatic cells migration in a ROS-dependent manner, which was modulated with NAC treatment during the labeling. At the same time, proliferation and invasiveness appeared to be impaired by quantum dot effects, probably by cadmium present into quantum dot structure, reducing metastatic cells capacity. As a perspective, this novel methodology has the potential to be used as a complete instrument to fully characterize early metastatic steps, along with the in vivo interruption of metastatic cells' capacity for proliferation and invasion. The latter mentioned, plus the capacity of monitoring cell behavior at short labs, might open a new window for theranostics in the future. Thank you very much for your attention and interest. Any question, comment, or proposal, I will be happy to answer through my mail indicated here. Thank you very much.